Okay, thank you, Philip, for that wonderful introduction. I'll start by saying I have no disclosures and funding from the NIH, Alzheimer's Association, ADCS, VA, and internally. I'd like to also start by saying a lot of this work is due to the very valiant effort of one of our students, Harry Huang, who last year received one of 6 student awards at the AAIC conference. So he worked really hard on this. I wanted to make sure I gave him a shout out. So as all of us know, Alzheimer's disease is a very important public health crisis. It is the 6th leading cause of death in the United States among Caucasians, and the 4th leading cause of death for African Americans. And what I really try to do in my research is focus on preclinical indices of Alzheimer's disease and try to design interventions to help people such that, you know, by the age you're 90 years old, we're able to shift this curve and you don't have full-blown Alzheimer's, but, you know, you might have, you know, MCI or, you know, something that isn't quite as horrific as severe dementia. So we really try to instill interventions that are practical, they're affordable, that people will actually take part in and like to do that. So we're going to be talking about some of the biomarkers that we take into account when we're trying to devise these interventions today. In addition to looking at in preclinical work, something we really try to do is take careful consideration about who we enroll into our clinical trials, because a lot of people, you know, won't go on to develop Alzheimer's or vascular dementia or dementia in general. So we really try to pick people to enroll that we think are at the greatest risk. And that includes women, it includes Black and African American individuals, people who have a family history of dementia, and then people who have the predisposition in the heart-brain connection. So things like people who have increased vascular risk factors, and also try to play on the clinical trials that can modify these Alzheimer's risk factors to try to change the disease trajectory. So something we really focus on in our lab is the Black and African American component of individuals, and trying to make sure that we devise interventions, both pharmaceutical and lifestyle, that are applicable to all populations. So we know that African Americans are more at risk for dementia than whites, non-Hispanic whites. And we did a study back in 2016 showing that African Americans are actually 64% more likely than adults to become afflicted with Alzheimer's disease. And I'm not going to focus on this, but, you know, we all know that the main hallmarks for Alzheimer's disease pathology is increased beta amyloid and tau in the brain. And what that translates to in spinal fluid is decreased beta amyloid 42 and increased tau. And there's been 2 studies now that have shown that African Americans and whites have different baseline levels of tau across the lifespan, across disease states, such that African Americans actually have lower or better levels of tau, as we're finding in spinal fluid. The first investigator to find that was one of my collaborators at Emory, and then that's now been replicated in John Morris's group at WashU. We're also particularly interested in markers of inflammation, because, as all of us know, beta amyloid and tau are not the end-all be-all of dementia, and even of Alzheimer's disease. So you know, we really think that both central, meaning brain, and peripheral inflammatory systems are not sort of passive players in this overall dementia story. So instead, we think that they play a critical role in disease pathogenesis, and that inflammation and these AD biomarkers really work synergistically to accelerate the aging and the disease process. And we know from the literature that IL-10, MCP1, and C-reactive protein, there's different levels of these biomarkers in Alzheimer's disease patients versus controls. And then there's a little bit of research if you're trying to look at markers of inflammation that differ between whites and African Americans. Most of that is done in the periphery. Hardly anything is done in spinal fluid. And so what we're finding is that there's different levels of ICAM, VCAM, and C-reactive protein based on the population of study. It's really hard to talk about, the role of whether it be, brain biomarkers of Alzheimer's or inflammation and not take into account the blood-brain barrier. So that's something we're also particularly interested in. As we all know, the goal of the blood-brain barrier is to keep the good stuff in and keep the bad stuff out. We also take into account that you have increased permeability of the blood-brain barrier with age and sustained disease like hypertension, diabetes, hypercholesterolemia. And the blood-brain barrier is comprised of these endothelial cells, which form the blood-brain barrier, which is covered in these red dashed basement membranes and also surrounded by periocytes, which I'm going to focus on today, which is this yellow region, and also astrocyte feet. And the role of periocytes is to regulate blood flow, to maintain vascular homeostasis. So that is a very important role for the blood-brain barrier. As of now, there really is a paucity of biomarkers to be able to look at blood-brain barrier function in humans in vivo. And recently, within the last couple of years, a novel biomarker has emerged called PDGFR beta, and it is a way of measuring. It's a soluble platelet-derived growth factor receptor. Is the long term. It's highly expressed in the periocytes, and it's shed from injured human periocytes when they're exposed to A beta, again, one of the pathological hallmarks of Alzheimer's disease. In 2019, Dan Nation reported that CSF, SPGDFR beta, and was correlated to regional blood-brain barrier leakiness in the hippocampus specifically in preclinical Alzheimer's. And then he went on to report that PDGFR beta predicted cognitive decline as defined by the CDR score. And that even though there were these blood-brain barrier changes, he found this independently in these preclinical Alzheimer's patients, again, independently of both beta amyloid and tau. So he posited this as a novel biomarker that could be a preclinical indice of Alzheimer's and vascular dementia in general. Scott Miners and Pat Kehoe and the Bristol group also showed that PDGFR beta was increased in Alzheimer's disease. However, they did find that there was a relationship between not beta amyloid, but p-tau and t-tau. So we wanted to take all of this information together, and the rationale for this study was to investigate the interplay between AD biomarkers, inflammation, and this novel blood-brain barrier biomarker in a cohort who was cognitively normal at baseline in middle age, and but also at very high risk for Alzheimer's and vascular dementia. We really wanted to focus on middle age because that's the time of life when inflammation and vascular complications, if left untreated, become problematic, and also the time that we know the Alzheimer's disease biomarkers begin to develop. We also wanted to make sure we included a large and overrepresentation of African Americans because as we will point out, I think some of these biomarkers are different among groups. So this study is called ASCEND. It is a 2-year longitudinal observational study. We measured individuals at baseline, year 1 and year 2. And today I'm only going to be presenting some of the baseline data that I've outlined. We enrolled 80 individuals between the ages of 45 and 65, and every person had a biological parent with Alzheimer's, which was validated either by our Alzheimer's Center or the dementia questionnaire. We had longitudinal annual lumbar punctures on everybody to collect spinal fluid, which we looked at beta amyloid and tau concentrations, CSF inflammatory markers, SPDGFR beta, and RAS markers, which I'm not going to be talking about today. We also took blood for E4 genotyping and plasma inflammatory markers. We also had cognitive testing and MRI, which again, I won't talk about today. So these are the basic demographics of our participants. We had 30 African Americans, 50 Caucasians. The average age was about 59 across the entire cohort. We had more of an overrepresentation in the African American group of women. We found that it's traditionally a little bit more difficult to get African American men as opposed to white men to agree to lumbar punctures, let alone 3 of them. So we worked very hard to do that. The only other difference was African Americans, everyone was pretty highly educated, but African Americans made less money, even though they were a little bit more educated than Caucasians, but everyone was highly educated. And as you can see, everyone was pretty healthy. Again, this is a middle-age, research cohort. So, blood pressure on average was about 126 over 77. So no major clinical incidents, and both African Americans and whites had about 50% E4 positivity with one E4 allele, which is consistent with other cohorts of family history. So the first thing that we looked at was the levels of beta amyloid and tau in both African Americans and whites. And what we found, consistent with the prior 2 studies in MCI and preclinical Alzheimer's and Alzheimer's disease, was a difference in the levels of tau in spinal fluid between African Americans and whites, such that African Americans had lower levels of tau compared to whites, which is good. So this is the 3rd study now to show reduced T-tau, total tau, and phospho-tau in African Americans compared to whites, and the first study to show this in a middle-aged healthy cohort. And I'll go on to implications about that a little bit later for the sake of time. We also looked at markers of inflammation. So this is kind of a busy slide, but at the top here, this again is separated by the inflammatory marker and then split by African Americans and whites. And the top is looking at plasma, peripheral inflammatory markers, and the bottom half is looking at inflammatory markers in spinal fluid. So we have some significant differences between African Americans and whites in these peripheral markers, such that African Americans had higher peripheral inflammatory indices in some markers compared to whites. But if you look just sort of at the overall picture of these CSF inflammatory markers, we see a flip. So, we only see that there are 3, significant differences in this relatively small cohort between these CSF inflammatory markers, these race differences. But overall, the white participants have higher markers of inflammation in spinal fluid compared to African Americans. So we thought that was very interesting. And those markers were MMP, IL-7, and VCAM. So the peripheral inflammation, I'll just touch on again. African Americans had higher markers of peripheral inflammation overall, specifically IL-7, MCP1, MDC, CRP, and SAP in plasma. However, in the CNS, African Americans had lower levels of MMP2, IL-7, and VCAM. So we don't know exactly what this means. The complexity of the inflammatory story in the periphery is difficult in and of itself, and then you add kind of the brain to this story, and it becomes pretty pretty complicated. But MMPs seem to be, have both good and bad mechanisms of action, such that the benefits would be neurogenesis, remyelination of axons, and adverse would be blood-brain barrier disruption, which is kind of where we're kind of going to go with this. Same thing with VCAM. It's involved in, it's a marker of endothelial activation. The second thing that we did, again, was try to look at this marker, this potentially novel marker of blood-brain barrier permeability, which is PDGFR beta in spinal fluid. And again, we look at differences between African Americans and whites. And what we see is that there is a relationship between African Americans and whites, such that African Americans have lower, which is better, markers, a lower marker of this, potentially problematic blood-brain barrier indice. So just for to try to look at more of a holistic picture. This is definitely post hoc, but we wanted to look at the correlation between the blood-brain barrier biomarkers and the inflammatory markers. And what we see is a pretty striking relationship just on some of these, but the ones that we do see a relationship are, were pretty strong. So we saw a relationship between PDGFR beta and IL-8, TIMP1, TIMP2, VCAM1, and MMP2. So again, many of these are involved in what we think would be blood-brain barrier preservation and endothelial function. So just to recap, African Americans versus whites. African Americans have lower concentrations of S-PDGFR beta, which is a potential novel biomarker for parasite damage in the blood-brain barrier. And also CSF PDGFR beta correlated positively with MMP2, TIMP1, TIMP2, and VCAM. And all of these markers are involved in endothelial activation and blood-brain barrier functions. So, although only 2 previous studies have shown this novel biomarker, together, we're thinking that, the 3 markers that we're finding relationships between, which is S-PDGFR beta, MMP2, and VCAM, those together, might suggest that African Americans have less blood-brain barrier disruption than Caucasians. So one of the last things that we did was look at the relationship to see if there was a correlation, as minors found, between PDGFR beta and T-tau and P-tau. And we, in fact, did find a pretty striking relationship, not with beta amyloid, but with both total tau and phospho-tau between both of those markers and S-PDGFR beta in all participants. So our results were consistent with his results in individuals with clinical AD. But again, these are younger, cognitively healthy individuals that are at high risk. So take-home messages and future directions. African Americans had lower tau burden compared to whites, and African Americans also had lower levels in 3 markers related to blood-brain barrier dysfunction, which was MMP2, VCAM1, and the novel PDGFR beta, which may suggest that African Americans have less blood-brain barrier breakdown compared to whites. And, we think these racial differences could lead to race-dependent diagnosis of different types of neurodegenerative illnesses, if that is in fact how we are going to define the illness versus a clinical definition. And what we're looking to do now, this trial is closed. It's been closed for about a year, so we have all of the data. So we're trying to look at the longitudinal changes in biomarkers, which again include indices of the renin-angiotensin system and cognition across everybody over a 2-year period. They've subsequently been enrolled in a second R01, so we will have about 8 years on everybody with the same biomarkers. And what we're really trying to do now is look at some of the MRI results to see if we can determine any differences based on ASL in conjunction with cognition. So I finish up. I went a little over, excuse me, but thank the members of my team, my collaborators, many of whom are on this call, and big thank you to Henrik Zetterberg, Scott Miners, Pat Kehoe, and Ruth for their hard work on this project. Thank you.